Our next speaker is Professor Adrienne Correa from Rice University, and she will be talking to us about challenges in coral reef research and conservation. Please welcome Professor Correa.
Hello, everyone. Thank you first to the organizers. It's been wonderful to be here. My name's Adrienne Correa, and I am a technology user from Rice University in Houston, Texas, US. And I'm a coral reef microbial ecologist, seeking to understand how microorganisms like these, hopefully, yeah, there we go. These dinoflagellates and viruses that infect them, all of which live inside of coral tissues, can influence coral health, along with things like reef fish. And so, one of the things that I want to bring up before I get started talking about this is that we've come quite a ways in understanding how microorganisms associated with corals, what they do and how they interact with each other and how they might influence reef health. We've learned a lot in the last 20 years, but there are a lot of things that we're still trying to understand just on the biological side. And so it really resonated what Michael Roberts and what Dave said in their talks about the fact that given the decreasing timeline that we have to protect reefs, sometimes we're having to start pick something and start trying to work with it, maybe before scientifically we would feel happy about having to do that. And certainly, as I have really been learning over the last day and a half, robotics and autonomous sampling can help a lot with making that happen for reef restoration. So we've talked about how the scale and complexity of coral reef environments can pose some research and conservation challenges. And, just to review, that's because reefs form these massive frameworks that stretch hundreds of kilometers that you can see from space. But when you zoom in, they're made up of these individual coral colonies building this giant framework. And if you zoom in even further, for example, onto one of these branches of a coral colony and look at the clonal polyps, which you can see here, that's the individual unit of the coral animal. It's microorganisms living inside of these coral animal tissues that are helping to fuel the production of these reefs. And so if we want to preserve or restore these massive reef frameworks to deal with some of these microscopic level interactions that are happening in individual coral colonies. And so one famous example within the coral world are these dinoflagellate algae. They're microbial symbionts in the family Symbiodiniaceae that live in the gastroderm of the coral tissue. And these have a typically beneficial partnership with coral animals in that these dinoflagellates photosynthesize sugars, and they give a lot of that sugar to the coral animal tissue, which helps subsidize the coral animal in building these giant reef frameworks. And then in return, the coral animal is giving nutrients to Symbiodiniaceae through its waste products, as well as a stable place to live in a reef environment. And this is so within Symbiodiniaceae, I'm not going to get into the details of this, but this is sort of a family tree of Symbiodiniaceae, and there are it's a very diverse group, and you'll see names like Cladocopium and Durastinium. These are all groups within Symbiodiniaceae. And one of the things that has been very interesting within Symbiodiniaceae is we've seen that some different genetic lineages within this group can have physiological differences in how they influence coral colony function. And so once we've realized that, we've started to care about, okay, not just what coral genotypes are present on a reef and how can we manipulate those, but also thinking about the genetic diversity of some of these microorganisms and whether or not we also should be trying to manipulate those within a coral colony or selectively breed, etc. Okay. So as I was just saying, for example, Symbiodiniaceae can influence coral colony survival and function. So as an example, folks have talked about how you can break these coral animals apart and then manipulate the different pieces with the microorganisms they contain. So for example, if you have a coral and you break it in half and some of it in a simplified example contains Durastinium lineage and another piece contains a Cladocopium lineage, you might find that the piece containing this Cladocopium lineage grows faster because these symbionts give more sugars to their host. But there can be a trade-off in that then if there is a heat wave driven by climate change on a reef, some of these Durastinium lineages will be better able to tolerate that change in temperature. And so you might have the symbiotic partnership within the coral persist despite this thermal stress with Durastinium dominance, whereas if other Symbiodiniaceae are dominating, a coral might bleach and potentially die. So when we are thinking about these sort of context-dependent trade-offs in how the microorganisms are influencing colony function, that's a moment when we also start to think about human interventions on whether or not we can sort of minimize trade-offs and increase symbiotic benefit. But it's not so simple as just thinking about Symbiodiniaceae. There are a whole bunch of different kinds of microorganisms that live in and on corals and influence their function. We know more about some of these and less about others. So bacteria, coral-associated bacteria is another group that we have learned a lot about, and I'm sure Raquel's going to be talking a lot about that in her talk. And then there are fungi and archaea present. And these not only are interacting with the coral animal, but they also can be interacting with each other and can have important relationships with each other. And then additionally, we're learning about viruses associated with corals and their microorganisms. This is something that my group works on that I won't have time to talk about today, but these different kinds of viruses can potentially infect each of these members of the coral colony. And often when we think about viruses, we think about them causing sickness or disease, and that certainly can happen within a coral colony. But there are other things that viral infections can do, and sometimes they can shape the composition of who else is present in a coral colony, and they may even sometimes have beneficial effects like transferring useful genes or other things. So this can get very complicated just on the biological side of thinking about what's happening and how we might want to try to work with that. So in terms of these, thinking about how we might intervene and use microorganisms associated with corals to boost their resistance to stress, this is an example of what one general workflow might be. So you could go to a coral colony and isolate and culture some of the microorganisms that live in it and try to identify which, pull out ones that are beneficial for the coral and try to grow those and then make a probiotic inoculum and then add that to corals on the reef to increase thermal tolerance or health, et cetera. And so there are all these different application goals, including perhaps accelerating growth of the colony, increasing survivorship, et cetera. And this is really important, but there are also challenges that we have to work on, such as we know that not all of the microorganisms that associate with a coral host are very readily cultured using the techniques that we have so far. So sometimes it can be difficult to pull things out and get them into culture. And then there's such a high diversity of all these microorganisms, we really don't know what all of them are doing yet. And then beyond this, getting to this scaling to the reef, and for example, dosing of probiotics at a reef scale, this is also very challenging. And you can think about in terms of efficacy, how would you need to apply these probiotics on the reef? Would you be able to just squirt them near a coral colony? Would you have to physically put them on the coral colony? Would you need to use something else to sort of vector them into a coral colony? And then another question is, even if you're satisfied that you're doing that part well, is a particular coral animal or a particular coral colony or species going to actually acquire or use those beneficial microorganisms in the way that you're hoping that they will? So these are all things to grapple with in addition to the scaling and delivery process. And so one of the things that I love to think about with my research group is the fact that these interactions with the microbes and corals are not occurring in isolation, not only in a coral colony, but also in an entire reef context, right? So out at Shusha Reef, there are going to be algae, like bushy algae. There are fishes that eat different things, like coral eating fish, or things that are grazing on algae. And so one of the things that we have been thinking about is whether or not we can use fish that bite pieces of coral typically without killing them, just biting a piece off, whether we can use that to, whether we can leverage that process that already happens in nature to potentially deliver some of these coral associated microorganisms around the reef. And then taking that a step further, is there some sort of robot or autonomous vehicle that we could use in a fish-like way to deliver microorganisms? So just very quickly, and I'm speaking to the technology developers, but I'm also sort of speaking to the biology folks here to try to convince you all that even though we haven't pinned down all of the pieces of this yet to confirm exactly what's happening, that this is a process that is perhaps interesting enough and promising enough that it's worth working on further. So just very quickly, on a reef, this is a very simplified sort of set of interactions, but corals and these bushy algae are fierce competitors for space on a reef bottom. And I just want to point out that in addition to corals having these dinoflagellates and bacteria on them, these algae also can have them. And some of the bacteria that are present on algal surfaces can be pathogens for corals. So they each have microorganisms associated with them. And then these coralivorous fish or coral eating fish will take bites of these corals. Some of these corallivores, they really mostly just eat corals as part of their diet. And then there are other fish, for example, grazer detritivores that will eat algae or make it easier for other organisms to eat algae, or they'll eat sort of organic dead matter on the reef. And then there are facultative corallivores that will eat some of both. So their diet contains some coral, but they also eat other things. And so something very interesting is that through the process of eating their different food sources, these different fishes are also consuming the microbes that these foundational reef species contain. And when they digest their food and then defecate as they swim across the reef, they are releasing these microorganisms either dead or alive across the reefscape. So this could potentially, for the coral eating fishes, be a thank you for biting kind of scenario where even though you might think of something that's biting and eating your corals as something that's causing a problem, and indeed they can break reef frameworks. And sometimes, for example, if you're outplanting small pieces of coral, they can be pest-like in that they might eat that whole piece of coral that you put out. At the same time, if you're thinking about an adult colony or a larger colony, there might be some benefits that occur. For example, if the symbionts are alive that they disperse in their feces, it provides an opportunity for neighboring colonies to take up and acquire those symbionts. Doing so might provide them with a different genetic type of symbiont that they can incorporate and establish. that could help them acclimatize to changing temperatures. And for an example, like where a bacteria from an algae ends up on a coral, it could even help algae sort of outcompete corals. So there are all these different kinds of ways that this could play out on a reefscape. And it's also interesting to think about on land as well for agricultural folks who are here. This can happen in other ecosystems. So for this to potentially be important, a couple of baseline questions are, do symbionts actually survive through the digestive tract of the consumer that eats them? And will the resource species, so if a coral is exposed to the feces from a coral eating fish, will it take up and acquire any symbiotic microbes that are in that fecal material? And I don't have time to go through all of this with you, and I don't want to get too biology focused anyway, but we have found in my lab that yes, there's strong evidence that many of these symbiotic microbes survive passage through the digestive tract. And now we're trying to figure out whether or not a recipient coral will actually acquire some of these symbionts and establish them. And also just to look at, from a nutrient standpoint and a physical material standpoint, what else might be happening with this fecal material in terms of how it interacts with a coral. Okay, and so just to the biology folks quickly, this problem of where are microbes on the reef and how many of them are there? This is for Symbiodiniaceae, and essentially the densities. This is Symbiodiniaceae cells per milliliter in either feces of different kinds of fish or in sediments and water, dead and live cells. And basically the densities are pretty low in sediments and water. And we've done this work so far mainly in Correa, French Polynesia in the South Pacific. And when you look at basically Symbiodiniaceae, both live and dead, in fish feces from these reefs, we can see that they are much higher in terms of density and that there are these live symbionts present. So potentially a better source for corals on the reef that need symbionts than water or sediments. And then again, these are Symbiodiniaceae genera, and I'm not going to go into depth with this here, but this is looking at whether or not the genetic identity of the symbionts in these different kinds of fish feces are similar to what a coral would actually host. And so here, darker blue colors just mean there's more of a particular kind of Symbiodiniaceae, and each of these is a fish feces or a coral genus. And one of the things that we found on these moraine reefs is that really and this is at the genus level, but it's also at a lineage scale as well. It's really indistinguishable, some of these coral eating fishes, their feces, the types of Symbiodiniaceae that are present genetically from what's predominant in some of these coral colonies. So they are delivering, and then this is looking at bacteria. So I've mostly been focusing on Symbiodiniaceae, but here we looked at bacteria present in some of these fish feces. And again, this is for an individual fish species going across a row. Darker green means it has more of a bacterial type, like a ASV, from either corals or these bushy macroalgae, or what's present in sediments and water. So what you can see is that in terms of bacteria as well, we've got a lot of this dark green color in the coral eating fish feces. So they have many of the kinds of bacteria that are present in corals in these coral eating fish feces, whereas these grazer detritivores have things that it's harder to see it here, but on these other screens, you can see more green with the algae. So certainly it's looking like the coral eating fish feces might be something that would be good for supplementing coral associated microbes across a reefscape, whereas the grazer detritivores have something different in their feces. And this is just getting more granular, looking at a particular, there are some beneficial or there are some probable bacterial symbionts like Endozoicomonas that we do see in the coral eating fish feces. And this Vibrio coralliticus is a known pathogen of corals. And we see using a higher resolution technique that there's significantly more of this in the grazer detritivore feces. There is some present in the coral eating fish feces, but it's less. And so I don't have time to show you. We have done some experiments where we take fish feces of these different types of fishes and we just physically place them on pieces of corals to see what happens, see what kind of health effects they might have. And the grazer detritivores are much tended to cause lesions or kill the fragment, whereas the coral eating fish feces had a much lesser effect in terms of that. And I can talk about that later if you like. So anyhow, it is certainly looking from these initial explorations, like coral eating fishes may, their feces may be sort of an environmental hotspot for microorganisms that are used by corals out on the reef. And so we started to say, okay, well, let's not just look at what's coming out of an individual fish, but let's try to look at this at a larger reef kind of scale. So we used some long-term data that had been collected from the reef, as well as some things that we collected ourselves, and basically just scaled and saw that this is an example of what we found for a few of these different coral eating fish feces. And we saw that in terms of scaling out to 100-meter squared patch per day, some of these fish feces, some of these fishes can move more than 100 million live Symbiodiniaceae cells. So this is potentially a large-scale process of coral microbe distribution that's already happening out on the reef, as long as you have corals present and as long as you have these coral eating fishes present. And from some of the, we did ground truthing sort of fish follows to make sure that the fishes were eating what we thought they were eating out on the reef, and to try to observe these defecation events and what happened to the feces. And it's different on different parts of the reef, but on the part of the reef that has a lot of corals, typically when a fish would defecate, you would, in 91% of the cases that we saw, at least some piece of the fecal material would land directly on a coral, which could facilitate transfer. And even when feces don't fall directly on a coral, there are processes such as water movement, et cetera, that could break up that fecal material over time and disperse those microorganisms in the vicinity of corals. So again, not tons of time to go into this, but we've been thinking essentially about how a coral-dominated habitat that has coral-eating fish in it, those coral-eating fish and that process that they do of eating corals and swimming around the reef and defecating might help kind of reinforce or condition that habitat to have a lot of these beneficial coral microbes present. Whereas if you have a reef, start degrading, you might have more of the grazing fish that eat these bushy algae, and you might tend to start having a feedback loop in which more and more of these potentially pathogenic, these microbes that are pathogenic for corals might be being spread around the reef. And so, you might have a tendency to sort of push one way or push the other way. And we can think about this in terms of management of some fish species like coral eating fishes that we might not typically manage for. We might want to start thinking about what some of these particular fish species mean out on the reef. So this is a very bizarre rendition of how we might think about using a robot. But basically, if there were a robot that could go around the reef and just suck pieces of coral polyp off, like break off a little piece of a branching coral or suck some polyps off of a massive coral, mush that up internally, maybe add it to some sort of synthetic vector that was negatively buoyant, like a fake fecal matrix, and then essentially just swim around the reef and release that, we might be able to have robots accomplishing this process that coral eating fishes accomplish, but perhaps even in a more targeted way. Like, for example, a robot is not going to come and eat your outplanted juvenile corals, but a coral eating fish might. And, thinking about if this were to happen, it would need to happen in addition to the targeted probiotic development that Raquel's going to talk about. but this could be an additional layer. And in terms of this additional piece, some of these, there would be different challenges, but some of these, like having to identify and culture the probiotics, you don't need to worry about with this sort of fish or fish-like system. And then, you know, sort of how you're applying the probiotics to the coral, you also don't need to think about as much, in that these feces would just be negatively buoyant and reach their intended part of the reef, hopefully. You know, some challenges that you might have are having your robot be able to recognize what kind of coral it should bite, and then to bite it appropriately and not ruin the colony. And then also recognize where to release these synthetic feces on the reef. You wouldn't want to release them over a sand channel or over a place where there's no coral. This challenge of, will the intended recipient coral establish and maintain the beneficial microorganisms, is still a question we need to work on. I think there's not a lot about this in the literature, in the published literature. There's nothing that shows that coral eating fish, the feces of coral eating fish, that a stony coral will establish microorganisms from the feces of a coral eating fish. But there are a couple of examples in the literature where an anemone has established Symbiodiniaceae from nudibranch feces or something similar, or stony coral larvae have been able to establish Symbiodiniaceae from giant clam fecal pellets. So giant clams also harbor Symbiodiniaceae, and they release Symbiodiniaceae in these fecal pellets. So I think these are very, very similar analogs that suggest that stony corals ought to be able to do this from coral eating fish feces as well. And I think I'm probably supposed to start wrapping it up now, but we're working on testing this in two ways. We're working on looking at whether or not coral larvae, so the coral babies that David talked about, can take up microbes, Symbiodiniaceae from these fish feces. And we're also looking at it from an adult coral perspective that I'll tell you about in a minute. So 50% of stony coral species are horizontal transmitters. That means that these dots are Symbiodiniaceae. That means that when they release their offspring during spawning, they don't have any Symbiodiniaceae present. So for these species, every coral that you see on the reef somehow managed to find or acquire these symbionts from the environment. And I showed you that these symbionts are low in density in the water and the sediments. So perhaps it is exposure to fish feces or being in the vicinity of fish feces that helps larvae do this. So we're working on this from a larval perspective, using horizontally transmitting species. And then we're also thinking about it from a bleached adult coral perspective. So bleaching is when the partnership between corals and Symbiodiniaceae breaks down due to increased temperature or light or other stressors. The coral tissue is still alive here. You can see the calcium carbonate skeleton through the living coral flesh, but the symbionts are gone. And then either a coral can recover if the stress goes away, or it can die and be overgrown by algae. So this is overgrowing algae in this scenario after a bleaching event, not that the coral is alive. And so we're thinking about whether fish feces might play an important role in helping adult corals recover their symbionts on the reef. And so we are trying this as well. And this might be really particularly interesting in moderate bleaching events where there's partial bleaching or patchy bleaching for different colonies on the reef. Sometimes there's a relaxation of coral preference by coral eating fishes during bleaching events. And there have been some studies shown that at least some coral eating fishes prefer to eat unbleached corals during a bleaching event. And it may be that these unbleached corals have symbiont types that are able to better tolerate high temperatures. And so in this kind of situation, although there are other sources of symbionts on the reef in the water or the sediments, it might be that during these heat waves that that the feces of coral-eating fish contain the most viable symbionts or specific types of symbionts that might be beneficial to corals. And so this could be sort of like a fecal microbial transplantation that occurs in humans. So this is another thing we're starting to test. And so just to sort of start wrapping up, biologically, we can think about coral-eating fishes and sort of an additional nuance to the role they might play on reefs and how we might use that maybe for some specific coral-eating fishes to try to bolster reef resilience to stress. And then we can, think about whether we can potentially bioprint some sort of fecal material and use it as a negatively buoyant delivery mechanism for a coral probiotic using robots. And so this is my user wish list. This one I have gone over. I think I am basically out of time, but I can talk with you, anyone who's interested on the break, about how another thing that would be really nice are robots that could go to specific corals that have been tagged and identified and sample repeatedly, especially at night, from all sorts of reef zones, and then pretty quickly get the sample back to you on land. A lot of times for biologists, it is not desirable for us to drive a boat out to a reef area, dive at night, and then come back due to safety concerns, both for ourselves and for the reef, because you don't want to hit the reef. But there are certain processes that you really want to capture in situ that are time sensitive. And so this would be very helpful. And then this is just further underscoring this need for repeated observations of environmental parameters, especially on remote reefs, in terms of things like changes in water quality after flooding, bleaching, disease, spawning, as came up earlier. And, some particular criteria are we need detection or observation systems that have a low failure rate, because, you can put these little hobo tags or other types of monitors out on the reef. And I've worked on reefs where a significant event happened and we went to go collect those data and the instrumentation had failed, even when you had redundancy. So this would be a huge help. And I'm interested in learning more about whether or not you could, have a docking station with a robot that when you say, okay, there's been flooding in the next month, I want the robot to start going out and sampling water quality. And then if you see that there's been a dip, maybe it can even go and sample the water and or corals. And then I just want to also point out that some of these for some of these things, if a robot is acting as a sentinel and monitoring, it's really going to need to be able to be quite discerning at times in terms of, what it's seeing on the reef, because when a coral's bleaching, as I mentioned, the coral tissue is still alive and it's still present on the skeleton, but on the calcium carbonate framework. But often you can't see it unless you can get really close up. With coral disease, a lot of times there's a lesion in that the tissue has sloughed completely off part of the coral and the coral's dead at that point. And sometimes, so some of the signs of certain diseases versus bleaching versus predation from a snail or something like that can look quite similar. And so to be maximally useful, if you're going to employ a robot to go and detect something like an emerging disease or something on a reef, you need it to be able to tell the difference between some of those tricky types of situations. And so with that, thanks to my team and funding sources. And I don't know if we have time for any questions, but